Today we're going to talk about Chapter 21, Part C, the respiratory system. First, we'll talk about oxygen transport. Most oxygen is transported in the blood inside red blood cells. Very little oxygen is going to be transported in blood plasma directly. Inside red blood cells, oxygen binds to hemoglobin, and when it binds to hemoglobin, that hemoglobin is called oxyhemoglobin. Reduced hemoglobin is hemoglobin that has released oxygen molecules. It may also be called deoxyhemoglobin. Loading and unloading oxygen are facilitated by a shape change in hemoglobin. As it begins to bind oxygen, it changes shape, increasing its affinity for oxygen. It does the reverse as it begins to release oxygen, changing its shape once again to decrease its affinity for oxygen. If a hemoglobin molecule has all four heme groups occupied with molecular oxygen, we consider that hemoglobin molecule fully saturated, whereas if it only has 1, 2, or 3 hemes attached to oxygen, it is partially saturated. Factors such as the partial pressure of oxygen, partial pressure of carbon dioxide, temperature, blood pH, and the concentration of BPG can influence hemoglobin saturation. The partial pressure of oxygen does heavily influence binding release of oxygen with hemoglobin, and this can be charted against that partial pressure. We can compare hemoglobin saturation to the partial pressure of oxygen and create a graph, but the curve is not linear. Instead, it's S-shaped, and this S-shaped or sigmoidal curve that's on a graph is referred to as the oxygen-hemoglobin dissociation curve. The amount of oxygen carried by hemoglobin depends on the amount of oxygen available locally. So we can see using the oxygen-hemoglobin dissociation curve that this relationship ensures optimal oxygen pickup and delivery. In arterial blood, the partial pressure of oxygen is about 100 millimeters of mercury, and at this partial pressure of oxygen, hemoglobin is 98% saturated. So at this saturation, even deep breathing is not going to increase oxygen binding much further. Whereas in venous blood, hemoglobin is only 70% saturated at a partial pressure of 40 millimeters of mercury. This is called the venous reserve because this is oxygen that's still remaining on hemoglobin in venous blood, and it technically can still be used. Here's more oxygen-hemoglobin dissociation curves. We can see that at sea level, that partial pressure of oxygen is still about 100 millimeters of mercury, like in those arterial systems that we saw in the last slide. And we're going to have a hemoglobin saturation in the 90s. And it's not going to change much even with a decrease in partial pressure. As long as that change isn't much, the top portion of the sigmoidal curve levels out, and we won't experience that much of a change in hemoglobin saturation, even with an altitude change. Other factors that can influence hemoglobin saturation are increases in temperature, the amount of hydrogen ions, the amount of carbon dioxide, and the amount of BPG. These, when they increase, can cause oxygen to dissociate from hemoglobin or result in a decrease of hemoglobin's affinity for oxygen. It often occurs in systemic capillaries, which allows oxygen to then leave hemoglobin and go to tissues. So what we're seeing here is an increase in these metabolites enhances oxygen unloading. And if we were looking at the dissociation curve, it would shift that curve to the right. Whereas if we decrease these factors, it would shift the curve to the left, and that would decrease oxygen unloading from the blood. So increases in these metabolites would increase oxygen unloading. Decreases in these metabolites would decrease oxygen unloading. So normal body temperature would be more likely to cause oxygen unloading, especially in tissues that are actively performing metabolism. So active or exercise hyperemia would bring warm blood to the area, and cells that are performing metabolism would also produce heat as a byproduct, and these increases in metabolites, including that of temperature, would increase hemoglobin and oxygen dissociation. Increased carbon dioxide would also cause dissociation of oxygen from hemoglobin, and here we can see that the increased carbon dioxide has shifted the curve to the right. BPG is a product produced by glycolysis, and when BPG levels are high, oxygen levels are low. And this is because cells metabolize glucose, so high BPG would encourage oxygen to dissociate from hemoglobin and go to the needy cells. The declining pH or acidosis and increase in carbon dioxide cause the Hb, which is the hemoglobin, O2, that's molecular oxygen, cause that bond to weaken. The weakening of the bond between hemoglobin and oxygen is called the Bohr effect, especially if it's because of an increase in metabolites and a decrease in pH in the associated tissue regions. But this ensures that oxygen unloading occurs where it's needed most. And heat production is also going to be in active tissues. And so when hemoglobin dissociates from oxygen in heated active tissues, this is also allowing increased oxygen unloading to those active tissues that need oxygen. Next, we're going to look at carbon dioxide transport. Carbon dioxide is primarily transported in the blood plasma as bicarbonate. You can see that some carbon dioxide is dissolved directly into the plasma. Some of the carbon dioxide is going to be chemically bound to hemoglobin. It binds to the globin chain instead of the heme group. And carbon dioxide bound to hemoglobin is called carbaminohemoglobin. But again, most carbon dioxide is going to be transported as bicarbonate ions in blood plasma. Therefore, there's a conversion step. We have to hydrate carbon dioxide, which is hydrated to carbonic acid, which is unstable and breaks down into hydrogen ions and bicarbonate ions. The bicarbonate ions then leave the red blood cell because this reaction generally occurs in a red blood cell where carbonic anhydrase is located. Carbonic anhydrase is an enzyme that makes the hydration of carbon dioxide a much quicker reaction, but that means the bicarbonate's made in the red blood cell and needs to leave to go into the blood plasma. And chloride is an ion that will hold its place until the bicarbonate is needed back in the red blood cell to be converted back into carbon dioxide. This shift of bicarbonate out and then chloride into the red blood cell is referred to as the chloride shift. That's what we're seeing here. Inside the red blood cell, the hydration of carbon dioxide into carbonic acid and then being broken down into bicarbonate and hydrogen ions happens quicker because of the enzyme carbonic anhydrase that is only in red blood cells. What we're looking at here overall is internal respiration or tissue gas exchange, where CO2 comes from the tissues, travels across the interstitial space, and then into the blood. We're also seeing that oxygen is leaving the red blood cell and then going into the interstitial space and to tissues. So during internal respiration, there is carbon dioxide loading to the blood and oxygen unloading from the blood. And here is that chloride shift. As bicarbonate was made, it moves out into the blood plasma, and a chloride ion takes its place to stabilize this reaction. And we can also see that when oxygen leaves hemoglobin, the hemoglobin will be stabilized by the hydrogen ion from the hydration of carbon dioxide, producing reduced hemoglobin. The opposite of internal respiration is external respiration, which occurs in the pulmonary capillaries and is also known as pulmonary gas exchange. It's the complete opposite. We see bicarbonate move back into red blood cells, so chloride moves out. We convert bicarbonate and hydrogen ions to carbonic acid, which then splits to form carbon dioxide and water. That carbon dioxide would then be unloaded from the blood or leave and diffuse past the respiratory membrane into the alveolus or airspace. So here is external respiration, it's the whole thing. The blood is over here on the right, the airspace over here on the left. Carbon dioxide is being unloaded from the blood and going into the airspace. Oxygen is being loaded into the blood. Oxygen will go into the red blood cell, combine with reduced hemoglobin to create oxyhemoglobin, and the leftover hydrogen ion will combine with bicarbonate that moved back into the red blood cell to form carbonic acid, which quickly is converted back into carbon dioxide and water, and that carbon dioxide will diffuse out of the red blood cell into the plasma and then into the airspace. The carbon dioxide and oxygen are moving through the respiratory membrane, made up of the capillary endothelium, fused basement membrane, and the alveolar epithelium. This process is occurring via diffusion, and again, we're looking here at pulmonary gas exchange or external respiration, and earlier we were looking at internal respiration or tissue gas exchange. The Haldane effect is just an effect that talks about carbon dioxide transport, and essentially boils down to the fact that carbon dioxide doesn't like to bond or bind to hemoglobin if it's saturated with oxygen. So the less oxygen that's on a hemoglobin molecule, the more carbon dioxide that can be carried by that hemoglobin. So reduced hemoglobin is much more likely to form carbaminohemoglobin with a carbon dioxide molecule than oxyhemoglobin. The chemical equation where we hydrate carbon dioxide into carbonic acid and then it splits to bicarbonate and hydrogen ions is also known as the carbonic acid bicarbonate buffer system, and it does help the blood resist large swings in pH. If the hydrogen ion concentration in the blood rises, that means the blood is becoming more acidic. So those excess hydrogen ions need to be tied up, and we can combine them with bicarbonate, and bicarbonate and hydrogen ions can form carbonic acid, and tying up those free hydrogen ions will lower the increasing hydrogen ion concentration and resist changes in pH. If the hydrogen ion concentration begins to drop, meaning the pH is becoming more alkaline or increasing, the carbonic acid can be split back into bicarbonate and hydrogen ions. The released hydrogen ions introduce more free hydrogen ions, and increasing hydrogen ion concentrations will bring that pH back down. Bicarbonate is also considered the alkaline reserve of the carbonic acid bicarbonate buffer system because it works to buffer hydrogen ions in the blood. Changes in respiratory rate and depth can also affect blood pH. So slow, shallow breathing causes an increase of carbon dioxide in the blood, or it causes carbon dioxide retention. This is because we see reduced ventilation during shallow breathing. But when CO2 builds up in the blood, it also results in a buildup of hydrogen ions, and an increase in hydrogen ions reduces pH. Whereas rapid deep breathing, that would cause a decrease in carbon dioxide because this would increase ventilation and increase the amount of CO2 that moves through the respiratory membrane into the airspace and then out of the lungs. Decreased carbon dioxide in the blood also decreases hydrogen ion concentration in the blood, and that would result in a rise in pH. So respiration and changes in breathing rate and depth can definitely play a role in acid-base balance of the body. And last but not least, we want to just talk briefly about the hypoxias. Hypoxia is inadequate oxygen delivery to tissues, and this can result in a blue color to the skin called cyanosis. And we are going to classify the hypoxias by cause. There is anemic hypoxia, ischemic hypoxia, histotoxic hypoxia, hypoxemic hypoxia, and carbon monoxide poisoning. Anemic hypoxia is caused by too few red blood cells or an abnormal or too little amount of hemoglobin in the blood. This could be caused by iron deficiency anemia or sickle cell anemia. Ischemic hypoxia is because of impaired or blocked blood circulation, such as that from a thrombus. Histotoxic hypoxia is when cells are unable to use oxygen. So even though the oxygen is present, they're unable to use it in aerobic respiration. And that could be because of a poison. One metabolic poison that does such is cyanide. Hypoxemic hypoxia is abnormal ventilation, usually because of a pulmonary disorder, especially a restrictive or obstructive pulmonary disorder. Obstructive pulmonary disorders and restrictive pulmonary disorders include things like fibrosis, emphysema, and bronchitis. Carbon monoxide poisoning, especially from fire, competes with oxygen to bind to hemoglobin. And it binds better, carbon monoxide does, to hemoglobin than oxygen. So even though oxygen is present, tissues cannot use oxygen because instead carbon monoxide is taking up all of the heme groups and hemoglobin. So victims in this instance would have headaches, they can become flushed, and untreated carbon monoxide poisoning can actually be fatal. All right, next time we're going to talk about control of respiration, and we'll look at different brain centers, chemoreceptors, and other reflexes that control pulmonary ventilation as well as other processes that we relate to breathing and respiration.